we move to our next speaker. Pedro Baptista he's an RI researcher, and he's head of the Organ Bioengineering and Regenerative Medicine Laboratory at the Aragon Health Research Institute in Zaragoza. He has authored multiple peer-reviewed articles in the field of organ bioengineering and regenerative medicine, including the generation of the first human heart, sorry, I was mixed with the first human liver created in vitro by tissue decellularization and cell engraftment. So today he's going to teach us a bit on how to grow organs on demand, liver on demand. So.
Thank you, Laura, for the presentation, and thank you everyone for being here, particularly after the great lunch we had upstairs. I hope you're still awake. I hope at least I'm not adding up to the sleep you might be feeling right now. I also want to thank the organization for giving me the opportunity to be here, particularly after the great trip we had to China last year in September, where we were actually able to see in loco the amazing features and construction and sites in Taizhou and in Nanjing, which we actually have a colleague here that will be presenting after me. But what I want to try to describe you today, it's a little bit what we do in the lab at EASE Aragon, and a little bit what have been carrying me through my career. And this is really trying to do livers for transplantation. And the reason why we do this is because the burden of liver disease, unfortunately, for the ones that don't work in the area, is growing around the world. And it's true that alcohol is still one of the main issues that our societies have to deal with, but there are other issues we are finally able to deal with, particularly hepatitis C virus infection, which we now are able to cure with the new drugs. We are still not able to deal well with hepatitis B virus. And then there's the growing incidence of NASH, or non-alcoholic steatohepatitis, which is actually probably going to be the biggest issue we're going to have in liver disease in the future, this together with hepatocellular carcinoma. If we compare these figures from all around the world, or for the Western countries, and we compare this with the burden of liver disease in China, the picture is not that different. Although, for the ones that are here and don't know the numbers, China is unfortunately the country in the world with the highest incidence of liver patients or liver disease. There are a few differences, and hepatitis B virus has a little higher incidence than hepatitis C virus, and the reason for this is that the vaccination programs implemented in the Western world around the end of the 80s, beginning of the 90s, was implemented in China a little bit later, and this is the reason why there's still a lot more hepatitis B carriers than the hepatitis C virus. Although there's also alcoholic liver disease, but NASH or NAFLD is also one of the biggest issues. And if you guys want to know the figure, and for a population of about 1.7 billion, it is estimated that 400 million people in China are probably having some sort of liver disease. And this is the reason why the Chinese government is actually trying to tackle this by investing largely in new therapies. So it's also, on the good news side, the country in the world with the highest number of clinical trials, most of them in the liver area. So most of them come and are sponsored from public institutions, but there's a little portion also that comes from the private sector. Although if all things go wrong, the definitive therapy for liver disease is organ transplantation. And in here, China has a growing transplantation program and donation program all around the country. But if you try to figure or read this number, which is about 20,000 up to the year 2011, so maybe double now, this is the number of liver transplants that have been performed in China since 2000. This is clearly insufficient for the number of patients that they have with liver disease. And since the trend is to grow and the failure of livers is really trending to grow, we actually need, or China, not only China, also other countries in the world, we certainly need other sources for livers that do not come from human donors. And this is where I started basically my research. And one of the things that allowed this to become a reality was a simple technique called organ decellularization that basically consists on taking an organ, in this case a liver, that once hooked up to a peristaltic pump can be pumped through detergent through the whole liver. So if this is a kind of a picture or a drawing of the tissue with cells and ECM, if you apply the detergent, so you wash away all the cells and you end up with only the extracellular matrix. Now, with this, you basically remove all the cells and you end up with this product, which is basically a decellularized liver. Now, we know that these livers, once decellularized, you shouldn't see any cells. We know that most of the vascular structures should be preserved, and this is a scanning electron microscopy showing a portal triad, but the interesting point here is that you see the ECM, but you don't see any cells on it. Also, we know if we inject, and if you can load the video, I would really appreciate that on this right corner right there, please. The video, please, if possible. There you go. Thank you. Go ahead. So what you can see here is that even after the whole decellularization process, if you inject a blue dye on the portal vein, the dye still remains inside of the channels. I call it channels and not vascular structures because at this point they have no cells. It's just the extracellular matrix. But it kind of shows you the proof of principle. And this is exactly what we now use in order to go a little further with our technologies. And we are also developing machines that are automated in order to decellularize these organs, in order to basically stream up the whole decellularization process and make this a little bit more easy and more reproducible in order to stream this up and scale this up towards human organs. Now, if we have the scaffold, so what do we do with them? This is really the critical part. Decellularization, as I showed you before, is really not that difficult. But putting cells back in, generating new tissue, vascular tissue, hepatic tissue, is really the hard part. So if we take this, and we had, or we use a bioreactor in order to have hepatic cells and vascular cells, what we have at the end of this is actually something that is not really transparent anymore. So you actually are able to generate hepatic tissue. And when you take a look into this hepatic tissue, and this is human hepatic tissue, you see that it secretes urea and albumin pretty much like the hepatic cells do, and they are actually able to form some sort of hepatic tissue. I'm calling it some sort because the organization of the cells in this is still not exactly what you see in human healthy livers. Although there are certain things here that show us that the liver progenitors that we are loading into these scaffolds are actually able to differentiate accordingly what they are needed to do. And here in green, you see clusters of cells expressing albumin, which are sided by cells that are forming these ducts in red, expressing cytokeratin 19, which is a hallmark for bile ducts. Here you can actually see that some of the cells also already became bile ducts. So this is quite important because at the time of making a liver, you cannot only load this with hepatocytes. So that's why in the very beginning, we started to load this with hepatic cells, hepatic progenitors, but also vascular cells. And one of the things we are doing now is also to automate the whole process. So this is the new bioreactor we have been able to build. And with this, you're basically able to control pretty much like some of the ex vivo normothermic perfusion machines that were talked by Manolo Doblare before, but in this case, not a bona fide liver or kidney, but a bioengineered liver. So the idea is the same, and pretty much the sensors you need to use are the same. So there's really no much difference here. You want to keep the cells alive and the organ viable for transplantation. Now, what we are able to do now, and this is, I think, one of the biggest breakthroughs we already had here in Zaragoza, is that we are now finally able to reconstitute the whole vascular network. And why is this so important? Well, the reason is that if you are able to bioengineer some sort of liver tissue, if you don't really have a completely functional vascular network, the moment you transplant this in, the blood will clot, and then the liver will fail. So this is the reason why reconstituting the vascular network on these large organs, large solid organs, is so critical to be able to transplant this into animals and furthermore into humans. And here, what you can see is that the formation of small diameter blood vessels here in green, these are medium-sized blood vessels and these are large-sized blood vessels. And this is, I think, one of the critical issues that will bring us forward. We are also working on the animal models, since if you generate a bioengineered liver, being that human or porcine, you also need a representative model, insufficiency model, that will show you if your liver is functional or not. So we struggled a little bit with this new model that we created with a partial lipotectomy and a portal calva shunt. But finally, after 15 trials, 16 trials, and this is science, we are now able to transplant native liver grafts using this model here. And the 2 pigs we've been able to do, one of the things we see is that the livers, once transplanted, they are actually duplicate in mass after 30 days. So this is the good news, and we are now finally getting ready to transplant our bioengineered porcine livers into this same model. More news by the end of the year. If money lasts. But this is what we are doing on the front of livers on demand for transplantation. But using this type of technologies, you can also bioengineer liver tissue, or liver organoids as we call it, as a human liver tissue surrogate. So here you use the same raw material, so which is your cellular scaffolds or a cellular ECM. And once you seed, you cut a little disc out of this, you basically load it with cells, you form a small 3-dimensional organoid that then generates or develops into hepatocytes and bile ducts, pretty much what you saw the other time. The only difference is that here is really a small, very tiny organoid that you can use in order to try to see what really determines the differentiation of the liver progenitors into bile ducts and hepatocytes, to try to do drug teratogenesis. And here, by using a notch inhibitor, you see that you don't generate mature bile ducts, or just by seeing that this particular ECM that we use on the organoids is more conducive to assure functionality of the hepatocytes, by seeing here more albumin secretion than in other types of matrices. And summarizing on this, so I think one single technology, which is organ decellularization and recellularization, really allows us to generate liver organoids for studying mechanisms of liver development, liver disease, drug discovery. Or if you apply this into other bigger organs, you can actually study organ transplantation, angiogenesis, vascular mechanobiology, etc. Same technology can be applied to other organs, and we're doing this with the urinary sphincter bioengineering, uterus in collaboration with IB in Valencia, or kidney engineering with Wake Forest. And just to finish, I think I already spared my time, I just want to acknowledge our funding agencies and the group at ESA Aragon, and our collaborators at University Hospital in Zurich, in UHC Chapel Hill, Wake Forest University, and also at IBB in Lisbon. This is the team that I actually have the honor to lead, and we are part of a much bigger group, which actually was the person that hired me here to Zaragoza, Dr. Angel Lanas, and this is actually the way we make it work, is that we are a more basic bioengineering group inside of a clinical group, and this is really the way we like to work, and I think this is what opens new horizons in science. Thank you.